Welcome everybody to Marine Biology Live. This is the sixth in the series that we've been running. I'm delighted to have Oliver Clarke talk today on microplastics, the exploration of potential image and methods. Oliver, through his work with Zeiss, has been a friend of the MBA for quite a while, I think, and has been involved in cell physiology workshops and the optical workshops. So he's well known to us, and we were actually talking about different events that we could be running. And Oliver, I think, volunteered to do a Marine Biology Live session, or it might have been arm twisted in. But it's delighted he can join us today and take us through the various options with imaging for microplastics, of which I think there's a lot going on at the moment. And I think Oliver's interested in our views as well. And are there other things that we can be looking at as we go? So Oliver, if you want to take it away, and we'll look at questions later. Remember, please, to use the chat functions for any questions you may have. Thanks, Oliver.
Thank you very much, John, and it's my pleasure to be here. And I just wanted to also firstly thank the Marine Biological Association for, as you said, twisting my arm. It's a real pleasure to be here. As I said, the subject of my talk is an exploration of potential imaging methods. The first thing I want to say is I'm most certainly not an expert in microplastics, although I do have a PhD in chemistry, and a lot of the work on microplastics is chemistry related as well as biology and environmentally related. But what I do have is a fairly good high-level oversight of microscopy techniques in a global setting. So I'm also not really a microscope specialist in any specific division, but I know quite a lot about the main things going on in those top level areas. And I think that's where a lot of the questions that I have had over the last couple of years come from on this topic. And as John said, a lot of it is some questions I'm going to pose to you as a community, as well as give you some the basic information. I'm aware I have a mixed audience, so some of you will be experienced imaging specialists in specific fields, and perhaps others have just had a very light touch interaction with microscopes or none at all. So I'm addressing kind of both of those. So let me move on to my first slide, and a somewhat ubiquitous question is, what are microplastics? But I think that what you notice when you look into the literature around microplastics is that that's not always an easy question to answer. And it's very important actually to be able to answer that question if we want to be able to identify, characterize, track, locate microplastics in our environment. For me, one of the big questions actually that comes up is around nanoplastics, and as nanoplastics are formed, maybe entering the environment directly in some nanoparticulate processes, but also being formed as a result of mechanical actions in the environment. The big question is, to what extent are those nanoplastics being integrated into the ecosystems, and specifically individual biosystems within organisms and the food web, the sediment, and also in the water column itself. And I suppose one of the key questions is it possible for us to characterize and understand some of these processes using imaging in a joined up way? Again, on my kind of just looking around at what are the key questions, as a novice in microplastics, I found the statement or the publication from the UN Environment Programme and the section on microplastics, where they do actually quite clearly say what the six main questions that we have in terms of knowledge gap. And clearly this is one organisation's point of view, but it is interesting that they tend to, or what they've highlighted here, a lot of these questions are questions that are only going to be answered by microscopic investigations of what's happening in our environment. So now we get on a little bit to the imaging part. So the microscopists among you will know the different types of microscope disciplines there are. The non-microscopists may not know. And essentially at the moment, there are, I mean, you can segment this particular technique into much more than 5 segments, but there are basically wide field microscopes. There are 3D fluorescence microscopes that generally are looking at single image planes using a sophisticated set of optics. There are electron microscopes, as the name suggests, doesn't use light, uses electrons. There are X-ray microscopes. And there's also a range of spectroscopic, or as I like to call them, spectral microscopes. So these are systems where the output's not necessarily an image, but is a spectrum, be it infrared or mass spec or some kind of other data set. I will, as John said at the start, this will be shared, so don't feel you've got to kind of take all this information on board at once. But I tried to do, from my perspective, an objective overview, and it was done relatively quickly, of what I thought were the sort of strengths and weaknesses of the different fields in amongst some of the challenges we've got. And I picked out, for example, particle size as being the way to separate out the challenge. Are we looking at millimeter, micrometer, or nanometer? And then very crudely, a length scale in terms of the particle location, so centimeters, millimeters, micrometers, in terms of the organisms, and then sediments and water. And I was also interested in how we characterize the particles. Are we talking about distribution, dynamic interactions, the chemistry involved, and so on. So I think we'll come back to that slide a little bit later, but the main message is, when you look at it, you realize there's not really one imaging modality that answers all questions on all length scales and in all situations. So we have to explore the possibility of a unified solution, which I will come back to a bit later. So I'm going to go into these one by one now. So the first, and probably one of the most common types of microscopy techniques you'll find certainly in marine biology, is dark field, which is a form of bright field microscopy. And the main concept behind dark field is to use a kind of cone of light, which allows you to view only the light that is reflected out of this cone and back into the optical path of the microscope. I remember as a child being in my grandparents' room on a sunny day, and they had a particularly dusty house, and I was always amazed at how I could see all the dust particles on the sunny day when the light shone in the window, and it was effectively collimated by the window, and I could see all these particles, but I never saw them anywhere else. And the reason I could see them is that the light, the very directional light from the window, was bouncing off of these particles against the dark background of my grandparents' room, and I could see them, and they were like stars on a bright sunny day in a dark room. And that is how dark field works, but in a microscope. And you have to make sure that you take a few things into account. You need a high dynamic range camera if you're going to get nice images. You also need to think about the vessel that you put the sample in. If you have something with lots of scratches and texture on the bottom of the dish, then you're going to also get interference you won't get a nice black homogeneous background, which is what people look for in dark field imaging. And you also need to realize that it's possible to do this on an upright microscope, but it's also possible now to do very good dark field imaging on stereo microscopes. Not so good on inverted microscopes because of the condenser orientation, and it's just not quite so good. But nonetheless, a very useful technique, and probably the one that most people encounter first when they learn microscopy in this setting. Okay, so I've jumped from a very simple type of wide field microscopy to the second topic, which is 3D fluorescence microscopy. And I've got 3 examples. And what I'm asking the audience to do here is to extrapolate a little bit and try and think about how some of these techniques might be applied to imaging microplastics. The good news is that we actually use microplastics to calibrate many of these systems. So tiny plastic fluorescent beads are very often used to make sure these microscopes are performing to the highest level. We'll see some of those beads in an image a little bit later. In this particular example, we're using a special type of detector. You can see this kind of honeycomb image down here. And what this is doing, this is an array of mini sensors that's imaging the light that's coming out of this imaging system. And normally in a confocal microscope, we'd have a pinhole aperture, and then we'd look at all of the light coming out of the pinhole and measure it. In this case, we can actually segment that pinhole light and pick the brightest, most intense part of it. So we can isolate individual hexagons within this array and get very sharp, high resolution images, but using very soft, gentle illumination, which is actually really important, not only for photobleaching, but also for phototoxicity in live organisms. And this comes from the ARIScan confocal, but similar results can be achieved through deconvolution using software. And the image there is a zebrafish brain, and what I really like about that is that you've got the ability to resolve these axonal filaments on a light microscope. Okay, I'm going to just start the video so it runs while I talk. I think one of the most impressive developments in light microscopy in recent years has been the manipulation of the illumination systems that allow us to get a new kind of image, and it's really about changing the way we illuminate specimens to illuminate more of the specimen in a certain image plane to make the imaging faster. And what that allows us to do is to be quite gentle with our imaging. So we can often image living organisms. In this case, a zebrafish embryo, which is GFP stained. And this has been done on an instrument called a light sheet. That's a commercial name. The generic name is a single plane illumination microscope. I'll come on to a little bit more about that when I talk about the next system. If I just move my slide on. A kind of development from the single plane imaging microscope, and you can see the SPIM illumination method here. And I don't want to get too much into the physics, but this is essentially the point spread function, or what the light's doing in that system. And there is that sheet of light that allows you to sort of scan through the specimen. Now, what was developed a few years ago is a new type of light sheet system called a lattice light sheet. And instead of having a single in a sense, light plane, there are multiple light planes arranged in a lattice. Now, I'm not a physicist, and it still fries my brain a little bit thinking about how this happens, or how they do this. But the end result is we can actually image a lot more of the specimen in a much shorter period of time and collect a lot more data. And although the image I've shown here on a webinar maybe looks a little bit uninspiring, it's actually many, many terabytes of the entire mouse visual cortex imaged at high resolution. It's just zoomed out. From the group of Eric Betzig, and Eric's become a bit of a legend in the microscopy community because he's one of the pioneers of this technology. Okay, so we're moving out of imaging involving photons and into the world of electrons, but not completely, because we're talking now a little bit about a correlation between light microscopy and electron microscopy. And I actually think that electron microscopy is the thing that almost all the other imaging modalities want to correlate to, because it gives you this fantastic depth of field and this fantastic resolution. It has challenges around sample preparation, for sure, because living organisms don't like high vacuums and high voltages. But by the same token, you do get this incredible image story coming out of these systems if you can handle the sample preparation. And I talked about microbeads for imaging, and what you can actually see in the middle there is right in front of your eyes these micron beads. So they're 5 and 10 micron polystyrene beads with a fluorescent compound inside them. And they're used to calculate these point spread functions in microscopes like confocal microscopes to check that the optics are orientated. That image was taken on a variable pressure system, the Sigma 500 VP, and I suspect that that's coated after it was imaged in the light microscope. So it's probably coated with a metal coating, but it's also possible to image these live cells under variable vacuum pressure without the coating there, so that you can keep them semi-hydrated. Another very common approach now is to use cryo technology, so to actually freeze the sample to stabilize it while you're doing the electron microscopy. And this whole world of biological electron microscopy has expanded massively in the last 10 years, where prior to that it was really just a pathologist's tool or a material scientist's tool. So very significant and very important in terms of correlation. Again, another electron microscope, this time slightly different. This is called a focused ion beam microscope, and what it's doing is it's taking a block of tissue and essentially milling back through laser ablation, through the focused ion beam. And these are gallium ions fired at high power at the surface. And what they do is they essentially polish very thin layers of the specimen. So you get this kind of, and as after it's finished one run, it images and you get this block. And the correlation with the XRM is important because it puts that very small block at high resolution into the context of a bigger structure, in this case, the mouse brain. And at Zeiss, we've developed a lot of correlative know-how in how to put those data sets together and give context to what sometimes seems a slightly alien world of very high ultra resolution, and bring that back to a kind of developmental biology question. Okay, let's move forward. So a similar thing now, but without the electron microscopy, we've now moved into X-rays. So we're now in an X-ray microscope. Now, anyone who's broken their arm or had a CT scan will understand that X-rays can be used to image in a non-destructive way through a volume. But what you may not know is it's actually possible to get nanometer resolution now with these systems. This is a micro scale system. And what it's doing here is looking at a 20 millimeter soft sediment core scan. Now, in my little trawl of the literature, I realized that actually it's almost as important to understand what micro and nanoplastics are doing in these sediment layers that play a very important role in the marine ecosystem, as well as what's happening in an organism or in a water column. This video wasn't created specifically to look for plastic particles, but it is on a very kind of micron sort of length scale, looking at particles within a core. I believe this one was related more to understanding more a mineralogist's or geologist's approach, but you can really see the individual particles and how we can segment and identify them. And that can also be correlated quite strongly with electron microscopy further down the line. So that's a system called the Versa 620. It's an X-ray microscope, and it comes also with the ability to be able to do soft tissues. So instead of the sort of piece of sediment, you're now looking at a scan. It's not a movie, unfortunately, but a scan of a zebrafish embryo. Also a developing chicken embryo. So you can see how that sort of first stage of the primitive streak sort of turned up around and created that first part of the gut. And then you've also got something from the plant world, which is a corn plant tip developing. So they're using a type of phase contrast in conjunction with the X-rays to give you this contrast effect in the X-ray image. And that's also very powerful because you can get quite a big contrast, and a lot of people think that the contrast between microplastics in the systems and the surrounding tissues in terms of density would be quite good and quite easy for you to be able to see them, but I still haven't seen an image yet of in situ microplastic in an organism imaged with a micro CT. But that's definitely something that, if it's not out there already, would be very interesting to see. And finally, moving up within the X-ray world now to the nano scale. And as I mentioned earlier, with the nanoplastics being probably one of the big concerns in this area, having the power to understand how they distribute and what they do within systems is very important. So you're going to need systems that can resolve them and find them. And in this case, slightly easier because titanium dioxide is much denser than plastic. So the microparticles are easier to identify. But if we can tune this system and optimize it with phase contrast, we stand a chance of being able to identify nanoplastics within a biological system. In this case, it's TiO2 in a protozoa, again imaged on a Versa 810. John, am I okay for time? I haven't actually got my watch on. It's 11:24. So.
Yeah, absolutely fine. Yeah.
Okay, good. The next one now, I move on to the fifth and the final type of microscopy, which is spectral microscopy, or essentially a microscope that has a spectrometer attached to it. There are lots of types, but I think the ones that will be important for microplastics are the ones that have Raman. Now, Raman spectroscopy is a derivative of infrared spectroscopy. So you're essentially looking at the effect of light hitting or radiation hitting a chemical system, and you're looking at the vibration on the movement, the energy of the bonds between the different atoms in those compounds, and how they vibrate and how they give back out that energy, how they emit the energy, is very indicative as to the chemistry of the compound you're looking at, and very, very useful for identifying plastics. And there's quite a lot of work being done already on relatively crude systems, I would say. By crude, I mean they haven't necessarily identified and characterized different types of plastics on a very small scale within organisms that have been harvested from the wild, as it were. But there's a lot of model systems being studied that seem to show that we could make that leap if we could find the right processes. In this case, this is an example of paper where they've identified the different constituent parts that make up a layer of paper. The interesting one there is the fingerprint of the polymer. So plastics work very nicely with Raman and show a lot of promise. This is an in situ system, so the Raman imaging is inside the chamber. So the correlation is very, very good because everything kind of sits on the same mechanical stage format. If you separate the two, it becomes more of a challenge because you need a special stage to coordinate with something called fiducial points, which are basically markers in the stage that let both microscopes know how to orientate the specimen. And this is a Sigma 300 system with the Raman on the side. It's also possible with light microscopy. So some of you may have heard of a company called Renishaw. They make a product called the Envia Contour. There are other Raman imaging microscopes around. This is actually a confocal light microscope, so I've talked about that earlier. And in this case, the image shown is a tile scan, and they are looking, in this case, at microplastics. I'd urge you to go onto the Renishaw website. Actually, they have some nice articles, if you haven't already read them, around the possibilities of Raman imaging and microplastics. And in this case, they looked at rainwater washout, and they were able to identify 4 main classes of plastics in terms of the chemistry of the Raman signal that was developed, and that's a 25mm scan. Okay, so I just thought I'd come back to the this slide again, because we've clearly seen lots of different modalities and across the Widefield 3D scanning electron, X-ray, and the spectral microscopy. And again, I think I just want to reinforce the point that I think as an imaging community need a strategy, if it's not already in place, to understand what role these different imaging modalities play in answering the question and closing that knowledge gap. And I have just proposed a kind of or I suppose made a graphic that in a way puts it together from my point of view, whereby if you along the bottom you have the sort of hardware architecture, Zeiss is in a nice position because we have these technologies all on very similar hardware. So the integration of the hardware isn't fighting against you. We have it all on similar platforms. So sample processing for Widefield, X-ray microscopy is very good at multi-scale distribution. 3D biological processes are really the remit of the 3D fluorescence microscopes if you're looking at those fast systems, soft imaging, live organisms. The SEMs, they're the nanoscale correlators, so they really bring different elements together. If you can get that working, they're a really good way to tie data together and make sense of very nanomicroscopical interactions. And then finally, the spectral microscopes give you the characterization. They give you the proof that this is a PET particle or a rubber particle or whatever. But all those things are useless without bringing together an element of scale up. And for scale up, we're going to need machine learning. So we're going to have machine learning applied to finding samples, identifying samples, and then analyzing the data, and also automating the sample handling. And again, that's something that at Zeiss we've spent a lot of time and money building up within our portfolio. So most of these microscopes can be automated, and almost all of them have some type of early stage machine learning function that can be applied to help you really teach the system what to look for on the search for these microplastics in situ from these environmentally recovered samples. And also in the model systems. And then from that scale up, we're going to create a lot of data. So connectivity and correlation is going to be very important. You must have open source workflow and correlation platforms, and we need to have the ability to connect this data very easily. And again, at Zeiss, we have a shared software architecture that makes this easy. I don't want to make this too much of a Zeiss pitch because it's a general topic, but that is a kind of an industry direction to get the acquisition and then the connectivity tied up in the same kind of environment. And then ultimately, if we're really going to solve the big problem of identified by the UN, which is making people understand that microplastics is a problem and to do something about it, then collaboration on a global level and communication is going to be very, very important. So that's the top of this kind of pyramid. Okay, so just a couple of points. There is a lot of funding around, and on behalf of Zeiss, I would like to get Zeiss much more involved. We are involved in a couple of programs that I can't talk about in this talk yet, but I would like to offer Zeiss UK as a conduit to get UK organisations involved in projects. We're actually in a good position at Zeiss to be able to offer proof of concept. We've got very good applications people, lots and lots of equipment, and we've got very good experience at working with academic organisations to write joint proposals, for example, to the EU. Innovate UK is also, I think, an important partner, certainly to fund some of the proof of concept studies. And obviously, you know, you all know NERC and their involvement and funded projects so far. Okay, so the door is open from my side. Contact me however you want after this presentation. And then finally, I shouldn't forget the most important job of all, which is to make sure that we carry on inspiring the scientists of tomorrow, not to make too much of a kind of cheesy buzzword. But I mean, I have a 7-year-old daughter, and she's just started learning about microplastics with me while I'm running this project, and I can tell she's really engaged, and she's learning about it in school as well. So I think we have to keep this dialogue going, and having a very strong STEM outreach message on the back of this, and trying to make it serious science, not trivial science for kids, and respecting that kids can take on board serious science is really important. And that's certainly a mission of Zeiss, and I'd be happy, and we're already engaged with the Marine Biological Association on some topics already. And that's the end of my talk. Thanks very much for your attention. I'm happy to take some questions, and hopefully I can answer them.
Thank you, Oliver. That was fantastic. Some amazing imagery there. I'm sure that others in the audience have incredible imagery as well, but fantastic to see it on the screen. A couple of questions coming in. A couple really on science communication, because we touched on before when we were talking with Maya before. Maya's asked, how do you think all of this fantastic technology and imagery produced help engage wider new audiences with microscopy and the microscopic world, or making people understand issues such as microplastics? So what's the best way?
Well, respectfully, I think, I'm not a fan of, so I'm involved in a lot of scientific communication. It's a passion of mine, and I'm probably the Zeiss specialist on that. And I follow kind of a guideline of respecting the science and respecting the people's ability to understand it. What you find is a very strong pull to try to have kind of whiz-bang entertainment-based science, and that's okay, but I'm not really sure what the shelf life of that approach is. We very much want to make sure that we respect the topics and give credit to people's, especially in this age range that you see in this picture here. Every time we go, this is from the Big Bang Science Fair, and whether it's parents in their sort of 30s and 40s, or teachers, or 7 or 8 year olds, they understand these issues. And, I don't think we should patronize them. We should get them involved. In terms of the really high end things, there's some really cool stuff. So for example, the augmented reality, virtual reality work that Zeiss has done with a company called Revis, where we use the Oculus Rift headset to take some of these data sets and put them into a VR environment. That's hugely powerful. So making sure we use the digital technologies that these digital natives are very familiar with, and keep it serious and respectful. Sorry, bit of a...
Okay. Are there locations where people can access some of the imagery that has been taken? Because obviously, people can't, most people don't have these systems themselves, but if the wider sort of membership was interested in how they could access some of the images, or are there any sort of banks of imagery available?
The short answer to that is no. In a way, the scientific community's become the sort of gatekeepers of perhaps some of the most interesting stuff that they've produced. And we could do a lot better. I say we as if I'm still a scientist, but I mean, we could do much better, and Zeiss is trying to take a more active role in doing that through outreach and public engagement programs. So yeah, okay. But I'd be very happy to talk to anyone who's got some cool ideas. We've got tons of images. We'd be more than happy to look at some material. Some of you might have seen Marty Jobson's videos on YouTube. He's doing a thing called Micro Minute, where again follows the same mantra, not silly science, this is proper science, but delivered in a way that 8, 9, 10 year olds can understand, and really good stuff.
Excellent. Okay, thank you. A question from Naz says, thank you for a fascinating talk. Some brilliant technology. If we wanted to test out and play with some bits of kit to see if they would work with our samples, what should we do?
Contact me is probably the short answer, because not everybody has an XRM in their back pocket. They're pretty big systems. There are a lot of them in the UK, so we can either give you access to our network of people who have these devices and have the expertise. There's a pretty big installed base in the UK on the XRM side. SEMs, we're quite lucky in that we have a centre of excellence. Here, I'm in the building where the first commercial SEMs, not this exact building, but Zeiss SEMs were first commercialised from Cambridge University 60 years ago, and some of the sort of stuff that still goes on downstairs owes its roots to that. So we've got systems here on the light microscopy side. So we've got, for example, Alex Sosic in the call. So from the Gurdon Institute in Cambridge, they have imaging systems, and I'm sure if you wanted to play around with imaging microplastics, that could also be arranged. And we also have the ability to send samples off to our demo centres around the world. So lots of options. You don't initially have to go and try and find £500,000 to buy a system just to find out that it doesn't work. So we do an awful lot of proof of concept.
Excellent. Yeah, there's been a couple of comments on that from Colin Brownlee from the MBA. Of course, the MBA itself has some really good imaging.
Absolutely, yes.
And,
yes.
So I think internally we need to talk to each other about using those. And Emmanuel Reynard said, I've done tons of plastic images during the Tara Oceans expedition. We have expertise in light sheet imaging of microplastics in the lab and soon underwater and possibly to do correlative work. So there's a lot of information out there. So yeah.
I'd be really keen to talk to that group about trying to look at microplastics when we get the lattice illumination set up and launched in a light sheet format. So the combination of those two technologies, I think, are extremely powerful, because that lattice light sheet technology is very fast, but it has that sort of cellular resolution, so you can really see very interesting dynamic processes without throwing tons of light at something and either killing it or influencing its behavior in a negative way.
Thank you. Really following up on the communication and the sort of imaging, accessibility to imaging, Leanne was asking, are there any recommendations or materials we could pass on to the young marine biologists category to further their interests? I think what you were saying is that not particularly, but it is something maybe we should be looking at in making materials available.
Yes, absolutely. I mean, the thing that young people consume the most are short videos, and I'm 45 years old, so I shouldn't know anything about TikTok. But actually, say what you will about TikTok, there are, funnily enough, quite a few people doing microscopy on TikTok, and of all the places I would not expect to see some quite serious science, there is some quite serious stuff being put out there. There's actually a lady from the US whose TikTok name is Microscopy, I think BEA, and she does all kinds of marine, she specializes in marine stuff. Her day job is a marine biologist at one of the big institutes in the US, and she puts videos up all the time, and I'm astounded that 40,000 people have liked it within half an hour and stuff. So we ought to make sure that we keep up with where people are consuming information and the age groups that we need to be targeting. I think that's just as valid a question as what do we give them to consume. It's getting the channel right.
Excellent. So Emmanuel says, happy to talk and to help with lattice, and various other people saying thanks very much. And yes, the MBA will have its own TikTok on the coming months, so perhaps we'll see you on TikTok and use that. Okay, if you could stop sharing your screen.
Absolutely. There we go.
And then I'll. So yeah, thank you, Oliver, for a fantastic talk, and I think there's things to follow up there. Just to let everybody know that the next event will be part of our TMB deep dive. So that's the Marine Biologist magazine, and looking into the importance of natural history with Fernando Barrero. So that'll be on Tuesday, the 10th of November at 11 AM, and I'm sure will be a very entertaining discussion. Linked to that is, of course, the Marine Biologist is now out, and members should be receiving that in the next couple of days. So the magazine is already out. So yes, look out for that, and then follow up with the TMB deep dive. So yeah, thank you everybody for joining. Fantastic talk. Please remember that if you've got a talk you'd like to put forward, or come forward with ideas or ideas on how we can extend the Marine Biologist, the Marine Biology Live, then please let us know and get in touch. The MBA is a membership organisation, so we're keen for people to carry on joining the MBA and getting involved with the organisation. Okay, so at that point I'll leave it, and just to thank Oliver again for a great talk. Thanks very much, everybody. Bye.